author of 25 publications in medical journals. Currently, he is a special volunteer at the Rocky Mountain Laboratory of the National Institute of Allergy and Infectious Disease in Hamilton. Before I introduce or bring Dr. Johnson up to the podium, keep in mind when there's a question and answer period here at the end, please use the microphone so other members who are streaming online are able to hear the questions. So please bring a warm welcome to Dr. Steve Johnson.
Thank you for that nice introduction. I have been thinking about genomics for a long time and have talked about it. I have this T-shirt that I got, I'm not sure what year, but it was on the cover of Science magazine in December of 2007. And at that time, I wasn't real excited that the Genome project, which had been through its first round of analysis, was getting anywhere clinically. In the last 10 years, however, it's been rather extraordinary what's been happening. And you'll notice, if you look at the handout that I prepared for today, so that you wouldn't be necessarily distracted by trying to write something down if you wanted to look it up later, then most of the references actually come from just the last 6 months, and in fact, a chunk of them from 2017. It's rather amazing what is happening. And the thing is that even though I pretend I'm Dr. Science, if you remember that from National Public Radio, I know more than you do, but probably not much. I only have a master's in science. That's the line from Dr. Science anyway. What I wanted to do is to actually concentrate on a couple of things. One is, how do you learn enough genomics that you can be a competent physician these days? How do you go about doing it? But the other part is that there are a number of ethical issues and also a number of practical things about applying it. So I'm trying to go over those today. This is not and could not possibly be in an hour a complete lecture on genomics. Now I'll see if I can run the slideshow here. So what are my motivations? Well, I wanted to be loyal to my genes and just let you know that that they're helping me with a lecture this morning. But I don't have any other conflicts of interest as far as limit my curiosity about genomics. I do have a lot of ignorance about the subject, and when Robin called a couple of weeks ago and said, boy, we have somebody that is dropping out on the 27th, would you be willing to give a presentation? I said, well, yes. And it occurred to me that I had 2 weeks, and I could spend a chunk of that time upgrading my knowledge of genomics, because that's something I've been meaning to do for quite a while. But it's pretty clear that getting yourself to a level of genomic knowledge that is going to be useful to you and your patients is not an easy task. And so it was kind of interesting to spend 2 weeks at it and realize that I had not gotten very far. Besides my ignorance, I had, for another reason, called a medical school colleague who worked at the NIH, and I hadn't talked to him in 45 years. I thought, well, I don't know how I'll get hold of him, but I looked on PubMed under his name and found that he'd written an article in 2013, and there was his email address. So I emailed him and got some very nice advice about working for the NIH. But I also noted that the paper that he'd written that I had just looked up was about learning genomics, and I thought, well, gee, I better really look at this. The thing is that when I go to the journal club, or when I am talking to fellow physicians or residents or medical students, I really depend a lot of times on a medical student to say, hey, you know, you didn't get this idea right, and here's what's really going on. The medical students, in my assessment anyway, aren't getting the kind of instruction that they need in this subject. And as a result, I thought it was worthwhile to at least get a little presentation on genomics here. And finally, I've had a couple of interesting patients in the last few months, or longer actually, that one has late onset cerebellar degeneration, another has facioscapulohumeral muscular dystrophy, and those turned out to be very interesting genomic problems. Okay, so there are more than a few philosophical issues that genomics raises, and I wanted to cover at least these four in this lecture one way or another. One that really is right at the top of the list, I think, is how much of your health really stems from your genome. And that's a question that I don't know if we can ever really answer, but there is some preliminary work, at least. You know, what's more important, nature or nurture? Well, that goes back a long ways, but actually, when we're getting down to more focused discussion, how much of the problems that we see in medicine are actually related to genomics. What constitutes informed consent? Because one of the problems with genomics is we don't know what we're going to find a lot of times, and we're sending off some blood or doing some analysis that may affect not only the patient but a number of family members. So we've got to be very careful to get informed consent about what we do. And another one that's really pretty philosophical, I think, is Faust. If you'll remember Goethe's play, he sold his soul to the devil in return for having a lot of knowledge. But should somebody, and you can substitute any scientist, politician, institution, or country for Faust, should they be allowed to sell their soul for the purpose of altering genomes? And finally, what will life be like when electronic medical records contain every patient's genome, and information technology algorithms and artificial intelligence black box programs take over health management? That one is, I think, a real upcoming issue. So about the first issue that I mentioned, I'll just give you a little bit of early evidence that just came out in, I think it was December 22nd, 2016, New England Journal of Medicine. Article was about genetic risk, adherence to a healthy lifestyle, and coronary disease. They had a total of 3 prospective studies that they put into this paper. So this is, in essence, something of a meta-analysis. The lifestyle factors that they picked out were that the patient was not currently smoking, was not obese, had regular physical activity at a certain level, and had a healthy diet. You had to have 3 out of 4 of those to be in the low risk group, and if you have more than 3 out of 4, you're either medium or high risk. The outcome of the study, when these patients were followed up, and the 3 studies were followed up for 10 years for whether they had heart attack or sudden death, favorable lifestyle was associated with a 46% fewer coronary events in the high-risk group. Well, that's pretty impressive since coronary disease is a leading cause of death. And if you have, if you could get a 46% reduction of the number of deaths from that disease, you'd be affecting at least 25% of the population. The problem with this study, of course, is that it's not a randomized trial. So whether you could actually get people to change their habits and have a favorable lifestyle remains to be determined, and it remains to be determined if you do an intervention on a randomized group, would that actually lower the risk as much. So here is a slide of the 3 studies. There actually were 4, but one was, the outcome was a coronary artery calcification, so I didn't include that part of the slide. But there is one group, and these are between 10 and 20,000 patients in each study, where they had, it was a study called atherosclerosis risk in communities, and you can see that with the low risk, intermediate, and high risk, and that goes across all 3 of these studies, that there's a dose response curve really as far as what happens with risk, but also what happens with the coronary event rate. So I thought that was really quite strong evidence that there is a real genomic issue here. I didn't mention, though, that they used 51 genes to set up a panel of what the coronary risk was. And the coronary risk was independent of the lifestyle factors. So you're really not dealing with something where a patient has a low risk because they inherited it, or a high risk because they, because they had the wrong genes. Those are independent of their risk factors that were looked at. Okay, the 4th issue of those 4 philosophical issues was something that I talked briefly with Randall Seacrest about yesterday, but there's a book review in Science Magazine on the 13th of January this year by a woman named Kathy O'Neill, and the title was Life in the Age of the Algorithm, and the author of the book in question was called, the title was Data for the People, How to Make Our Post-Privacy Economy Work for You. Well, it turns out that Andreas Wiegand was the chief scientist for Amazon. So you can imagine that this is a business book. But one of the things that he apparently emphasized, and the book has just come out, was something called an honest signal. If you're in marketing or in many areas of social science, an honest signal is one that you deduce from a person who tells you well, hey, I've always got good habits. I've never shot anybody in an episode of road rage or anything like that. And then you have some other evidence, an honest signal about what they really do. And anyway, the conclusion of the book review was that Wigand has written the ultimate guide to making things better for engineers and data scientists, but not so for the average person. And I think there's some real irony there, because the honest signal, at least according to the reviewer, would be that he has no clue about people. He's just an engineer who has no idea about what's going to be acceptable. And sometimes I think that might have to do with our electronics record system and physicians. Okay, my classmate from medical school, Eugene Passamani, had some ideas about learning genetics and or genomics, and he published this article 4 years ago. The thing is that it seemed to me, from my observations, at least at the Journal Club and with residents, is that what he said in the article is just as relevant right now as it was 4 years ago. And one of the first things he said is that building a knowledge base in genomics is much more difficult than incorporating incremental advances in established medical practice. And that's what we often get a lecture about at rounds is, hey, here's how we're coming along with a narrow part of medicine, and you can get the idea and you can use it if it's something practical for you. But when you talk about genomics, it's very broad. The example, article was in Clinical Pharmacology and Therapeutics, and the example would, of course, have something to do with medications. He had some polls of physicians indicating the vast majority of clinicians believe that individual patient drug response depends on genomics. I think that it was at least 80%. But very few clinicians, it was like less than 10%, had any medical school or graduate training in pharmacogenomics. One of the only actually figure in the article that Passamani wrote is this one about the cost of determining a genome, and I updated the graph from the National Institute of Genomics website up to 2015. It looks almost like the first quarter of 2015, and at first the cost went along with Moore's law, which is that you can get roughly twice as many transistors on a chip, or twice as much memory on a chip 2 years from now as you could this year. So if you notice that just as a logarithmic scale, but what's happened is that the cost of doing a genome with the automation that's taken place has got to be very cheap. And Abe Elias, who is a person I would recommend to you if you have a genomic question, he was briefly here as a family physician in Florence, but he works at the Shodair Hospital, and one of the things that he pointed out to me was it was getting so cheap to get a whole genome done that that Athena Diagnostics, where you look for just a group of genes or a single gene, has turned out to be far more expensive than looking at the whole genome. And so I think that it's safe to say that in the next year or two, we're going to be ordering genomes on patients by sending them to the lab. Anyway, how Assamani said was that there was no parallel cycle of improvement to parallel the drop in cost of the genome in physicians' ability to interpret it. The strategy paper from the National Institute of Genomics was that it seemed within reach that we could get there as far as going all the way from base pairs to bedside in the human genome. And that would be really what the Human Genome Project was trying to achieve. But it's going to take a while yet because I think we're, as practitioners, slower than expected. Okay, so I just thought, and this has no real basis in numbers exactly, but who are the heavy users of genomic analysis at this point? It seems to me that the human genetics people, even though there's very few of them, are the ones that use it the most, but oncology uses a huge amount of genomics to analyze tumors and also to help determine what drugs might help control a tumor. There's an awful lot of neurologic and pediatric disease that is pretty strange, and it's very nice to be able to have a genomic diagnosis supported if there is one. And then clinical pharmacologists have been working on this for quite a while. But who needs to use it that isn't as much? Preventive medicine, general medicine, algorithm designers, psychiatry for sure, and I think critical care and anesthesiology, because they could prevent a lot of problems if they could get a quick genome done and know what kind of a hole they're about to step into if they use the wrong medication or the wrong intervention. So what constitutes genomic medicine? Well, your handout has these websites on it, but they're really very nice information from the National Human Genome Research Institute, but also don't forget the Wikipedia. They have a really good article on genomics. So each person's genome comes in several subsets, and you can divide these up into nuclear, in the nucleus of the cell, mitochondrial, in the mitochondria, and microbial. And you probably wouldn't really think that your microbiome is exactly distinctive of you, but in fact it is. Whole genome analysis will be routine, I believe. Analyzing a single gene or a small panel of genes is getting to be too expensive, and you don't get the information that you would if you did a whole genome study. You do have to have a good idea about informed consent before you embark on this. So what do these terms mean? If we talk about sequencing, it's really like cutting and reassembling a ribbon. And the ribbon of DNA in each of your cells in the nucleus is almost 6.5 feet long. So you can't just analyze that with all by itself and get it right. It might be tied in a knot or something. And so what you do is cut it into smaller pieces, analyze the pieces, and then look for overlapping parts of the pieces that you cut it up into to determine how to put it back together. And that's a very tedious process, but it's pretty much automated now. Bioinformatics is, I think, for clinicians, the more interesting part of this. And it's a branch of biology that's concerned with acquisition, storage, display, and analysis of the information in the DNA sequence and the protein sequence data. So what about relating the genotypes to the phenotypes? Well, we've discovered a whole bunch of genes, and the pattern of inheritance, though, has been lagging because it's more complicated than we thought it was, and also because a number of genes that have been found that might be related to a disease, we don't know for sure if they are, and we don't even know what those genes do. So we have quite a way to go, and it takes a lot of investigation to clear this out. But we can remember from Mendel's studies, or the things that we learned in high school or whatever about dominant and recessive inheritance and X-linked. It's interesting to think about dominant inheritance, what actually is happening. There is something called haploinsufficiency. That is, you've got to have 2 genes to get the amount of action that you need. And if you only have 1, then that's not enough, and you have some symptoms. That's relatively uncommon explanation. There is something called dominant negative, and as best I could figure out, that means that the faulty gene is producing a product that interferes with the product from the good gene, and you end up then with just one bad gene that's really causing trouble. You can have a gene product that's directly toxic, which is very similar to this idea, and there would be other mechanisms too. X-linked, you're not getting sufficient gene product because you only have 1 X chromosome that's active, and if the mutation is on that X chromosome, then you're not going to have enough gene output to be effective, and you get symptoms. Recessive is autosomal recessive. You'll need usually 2 genes that are mutated in order to get an insufficient gene product about. And that means that recessive inheritance it requires a lot of people to have 1 bad gene and no symptoms before you get somebody with symptoms, because they end up with 2 bad genes. Finally, there's multi-locus variation, and that's where we need, if you have more than 1 gene that's required to get symptoms or signs of a disease, then we really need a lot more data to follow up with what is actually the connection, which genes are involved. And then there's some interesting work with Prader-Willi syndrome and Beckwith-Wiedemann syndrome. And I'd heard of these as a neurologist, but I hadn't really I've seen Prader-Willi, but I hadn't seen a Beckwith-Wiedemann until Dr. Beckwith came to my office. And I was surprised to learn from him that he was born in St. Ignatius. He's a pathologist. He's well into his 80s. And then Beckwith Avenue in Missoula is named after his family. So it's a small world. But in any case, this idea of imprinting is, I'll mention a little bit more, but it's really interesting way that genes are modified and passed along by methylation. So careful individual analysis for the patient and by the physician really is needed to figure out a therapy, and there are some. So what happens with Beckwith-Wiedemann? Well, I'll give you a quick view here, but there is a whole set of genes that are repressed. And you can think of the X chromosome. Every woman has 2, but at random, one of them is repressed. If you had both X chromosomes running, it's fatal. You're going to have an overdose of a ton of genes that are on the X chromosome, and you just don't survive that. So these other genes that are subject to that are inhibited or imprinted so that one of the genes doesn't work, there's about 100 sites across the non-X chromosomes in the body. And so what happens is, if one of those genes is not repressed, and you've got both genes active, as is shown down here, then you get an increased level of that gene product. And in the case of Beckwith-Wiedemann syndrome, you get a giant kid. You also get certain kinds of tumors. And it's taken a while to figure this out, but the imprinting is very similar to what happens on the X chromosome as far as it being repressed. And this is it's something I think a lot of us probably weren't aware of. All right, so if we go through the kinds of, or the sites where DNA is found, the human nuclei normally contain 23 pairs of chromosomes, and each parent contributes one chromosome to each of the pairs. The chromosomes consist of DNA as exons, which are genes, and they're interspersed with introns, what's between the genes, which are mostly control elements. And then there's also non-DNA support and control proteins that are part of the chromosome. And normal variants of exons are called alleles. And if there's a variant that is spontaneous and causes trouble, it's a mutation. So diseases can arise from abnormalities of the entire chromosome or part of it. Some of it can be deleted, for example, and encompass a number of genes that are missing or duplicated, such as trisomy 21 or there's a trisomy 18, I think. So that's extra function from those genes. Or there can be exon abnormalities, intron abnormalities, which alter the signaling inhibitory RNA, and then epigenetic mechanisms. One of the ways that we can answer the question about why we have just about the same number, 21,733 genes in a worm that's about 1 millimeter long, and we only have about 25,000 genes, is that we kind of mix and match the exons to make longer or shorter proteins that then have different functions. So there's a huge number of combinations of exons that can be used, and that explains why we have can make do with just a relatively smaller number of genes. This is called alternative splicing. That's what's took this off of the National Genome site, but there are other explanations also for, besides splicing, that would explain why we get more usefulness out of our genes than you might think by simply counting them. In any case, a lot of the difference between humans and worms, though, is also, or about the vast majority of it, is because all the introns that we're using to control what our genes do. And so we have a whole lot more DNA, but not so many extra genes. Mitochondrial DNA, mitochondria originated from intercellular bacteria that got into cells early in evolution and decided, hey, we can be the ATP producers for the cell, and so the cell benefited, and so did the mitochondrion, or so did the bacterium that was in there by having a place to live. The mitochondria are located outside the nucleus. They have their own DNA, and it's derived from the cytoplasm of the ovum. So inheritance of mitochondria is matrilineal. In most human cells, there's considerably more mitochondrial DNA than nuclear DNA because human cells contain hundreds to thousands of copies of mitochondria per cell. Well, that tells us, well, and we can deduce from that that when you get your DNA sequence, they combine the mitochondria with the nuclear when they're looking at it, and so you have to sort that out one way or another afterwards. Mitochondrial diseases are unpredictable because of the relative numbers of mitochondria that are a little bit different from one another. Their alleles are different, and so they can be really devastating though, because if you lose your ATP production, your cell can't survive. And then there's a personal microbiome. And the evidence at this point is that you can actually look at a person's microbiome and see the relative frequency of viruses and bacteria in the microbiome, and the types of microbes, and come up with a fingerprint. So it's pretty unique to a given individual. And one obvious place where the microbiome can restore health is if you're treating C. difficile diarrhea, you may have to do a fecal transplant, and that's quite successful for helping you get rid of your diarrhea. But the implications of the microbiome in health and disease are not real well worked out. That's pretty new information. What about the pharmacogenomics that Dr. Passamani talked about? Well, one that he didn't talk about is multiple chemical sensitivity. And I think you all have had the experience of the patient that comes in and says, well, I can't take this, I can't take that, I can't do this. And this one patient who I had been following for years for epilepsy and could only find about one medication that she could tolerate, said she had her P450 enzymes checked, and the reason that she had the problem tolerating medication was because of her metabolic genome. And the interesting thing about this is it actually appears to be the case, and there's an article about that. Codeine, some children and adults are rapid metabolizers of codeine, and if they take some codeine, then it's rapidly converted to the active narcotic, and the children could have a cardiac arrest, respiratory arrest after tonsillectomy if they were given codeine. Warfarin genotyping reduces hospitalization rates in a large study, and carbamazepine, there's a test that you can do for hypersensitivity, and about 5% of people have hypersensitivity that can be very severe. If you do the testing for HLA A3101, you can predict that. Now here's another illustration of using genomics for medications, and that's chemotherapy. And there's an algorithm that was published a couple of years ago, and the basic idea is that, and I'm not going to go through all of this because it's too detailed, but you look at a number of drugs and see what they have in common as far as their structure, how they work chemically. And you also look at the genome, and then you look at the interaction, and you can come up with some groups of drugs that would actually work for a given tumor. So you've done a genomic analysis on the tumor and seen which genes are activated by the tumor itself and also repressed by chemotherapy. Anyway, you can come up with a heat map like that. The authors here had about 75 different drugs that they looked at from the database, and they had 624 patients. And you can see that the green means that the drug was very effective in acting on the tumor, and the red means that the tumor was relatively resistant to that drug. Well, things are all over the map, and if you could predict what is going on from an individual, you could sure pick out the right drug as an oncologist, and then you'd have a chance to maybe actually end up with a cure in some cases. There's something also called a cancer immune set point, and this just was in an issue of Nature that it's a little hard to get a good picture from. I tried, and you'll see in a second, but the set point is a balance among factors that promote or suppress anti-cancer immunity. Cancers are related to somatic mutations. They're different from your other cells. Your immune system should be able to get rid of them, but a lot of times it doesn't. Is it just overwhelmed? Is it suppressed? What's going on there? So the set point actually is determined by a number of things: the antigens in the tumor, your personal genome, your age, which is you have more opportunity for somatic mutation, personal microbiome, viral infection, sunlight exposure, which has to do with the amount of vitamin D in you, and also certain tumors are, such as melanomas, are indirectly due to sunlight. And then also medications that suppress or activate immunity. So here's the fuzzy diagram from Nature, but what's really happening here is that if you're tolerant, then your immune system is suppressed, and if you have a lot of immune action, which has to do with inflammation, then you are going to combat the tumor. Well, how do you shift yourself from being here and having the tumor take over to here and getting rid of the tumor? And it's those factors that I just mentioned. That is your own genetics, your age, your microbiome, viral infection, exposure to sunlight are some of the factors involved. Okay, so one of the things that I wanted to do was talk about a couple of patients, because I think they're good illustrations. The first is a 68-year-old man who has ataxia. He uses a ski pole to keep his balance while he's walking, and he has a jerky eye movements when he's looking around. It's called saccadic pursuit. It was pretty clear to me 9 years ago when I first saw him that he had a cerebellar degeneration. And the symptoms have progressed over about 15 years. His mother had similar symptoms. She lived to be 94. His son had a malignant tumor at age 9, but he was cured. But then he died of a different malignancy at age 33. So we have, if you look at this, something happening in a dominant fashion, according to our usual way of looking at generations and seeing that there are symptoms. Abe Elias, who I mentioned is at Chaudere in Helena, I consulted him. He got some genetic studying done, and it turned out to be abnormal. So what was it? Well, the patient is hemizygous, meaning he has one probably bad gene, but not two. And the gene mutation actually has not been previously described. But the gene TDP1, I had to look it up real quick, plays an important role in repair of another damage from topoisomerase to DNA. So it's a DNA repair gene. And mutation of the human TDP1 gene has been reported in recessively inherited human cerebellar degeneration and axonal neuropathy, or SCAN. And also, it's mostly written up in what you can pull up on PubMed as having to do with response to cancer chemotherapy. Well, I was able to locate a researcher at St. Jude's who's been working on this gene. I got in contact with him, and he was really intrigued by the family history. And so we're going to investigate the blood sample. Whether we can find something that will benefit the patient is not entirely clear at this point, but I need to have the IRB and the informed consent state that we don't know exactly what we're going to find, but it could benefit the patient or his family. The researchers intend to do a tissue culture that includes that mutant gene and see what happens to see if the gene really is relevant to the symptoms. But you can imagine if you can't repair your DNA that you could end up with a higher chance of cancer, and that you could also end up with some problem in your nervous system, such as neuropathy or problem with the cerebellum because you can't repair your DNA. Now, facioscapulohumeral dystrophy is a different thing, and it's quite interesting how that's inherited. A 23-year-old woman had slowly progressive scapular winging, weakness of her face and arms, mild retinal vasculopathy, and hearing loss. Those are all distinctive features of FSH dystrophy. And she had been previously confirmed to have a reduced number of D4Z4 repeats in her 4q35 region in chromosome 4. The lack of repeats and the presence of only type A, at the end of this region there is a type A and a type B section of the region. If there are only type A distal alleles on one chromosome, and you have one chromosome that doesn't have enough repeats, you end up with the DUX4 gene not being repressed. Normally this gene is active during development early on, and then after that it's only found active in testicular tissue. Well, here's a woman. So how did that happen? Well, it wasn't repressed as it should have been, and so she got the symptoms. The DUX4 is toxic to muscle. So if that's not repressed, you get a relatively small polypeptide that is toxic to your muscle. And the inheritance pattern most often appears to be sporadic. You'd think it would be dominant, but because you also have this type A and B, you end up with, even if you have relatively few repeats, you end up with just about 1 in 3 or 1 in 2 chances that, that the disease is going to manifest itself. And right now there is an idea that we can have a therapeutic trial for this disease using either a small molecule or an inhibitory RNA. And this is just just shows what happens if the amount of D4Z4 is insufficient to suppress the excuse me, the gene region. You end up with a toxic protein. So what about gene therapy to wrap this up? Well, counseling and support have been the traditional things that we've done. And I think a lot of people don't really, a lot of physicians don't think, well, what's it worth to get a genome analysis on a patient? I mean, we can't do anything. Well, number one, that's wrong. And number two, you got to think about the family, because in another 10 years or 15 years or next month, there's going to be a treatment for a number of these diseases, and you want to know exactly what's going on. Anyway, counseling and support are very important because you can make the patient have a better quality of life, and you can also get some support for the family. Small molecule therapies, such as what I was talking about with the facioscapulohumeral dystrophy, is a possibility. And the mitochondrial donation is something that has just gotten started and was reported in the New England Journal at the end of December from Britain. And Brits have approved it. What you do is you have a 3-parent offspring. You take the mother who has bad mitochondria and you grab out her nucleus that has most of her DNA in it from her ovum. And you then you take a surrogate mother and you take out the nucleus from her cell, but leave the mitochondria, and you put the defective mitochondrial mother, you put her nucleus into the surrogate who has good mitochondria, and you have the cell fertilized, and you end up with a baby that doesn't have a mitochondrial disorder. I mean, that's pretty stupendous surgery, you know, to get down to an ovum and actually be able to switch nuclei. You couldn't just switch mitochondria. That would be much harder. So you just switch the nuclear material in the cell. Oligonucleotide therapeutics and also something called CRISPR are just on the cusp of being approved for human use, and those last two are the most exciting, I think. So what about oligonucleotide therapeutics? Well, I don't expect you to look at this whole diagram because it's kind of hard to see. But this was just in the New England Journal this year, 2017. And basically what's happening is you get some RNA in this particular case, and you have something that you attach to it so that it gets into the cell. Then the RNA and the sort of the adjuvant that gets you in there are broken down in the endosome. The RNA escapes and can get into the sarcoplasmic reticulum or wherever it finds the proper messenger RNA and cleave the cell and cleave the RNA, and then that whatever that protein is isn't going to be used. Well, in the case of and how does it happen then that this RNA knows exactly where to go? Well, it complements DNA, and so you can design an RNA that just exactly fits where you want to clip the other RNA in this case, but you can also have it clip DNA. So that's the basic idea, to infuse an inhibitory RNA and latch onto a specific messenger RNA, inhibit gene expression. And this was just reported this year in the New England Journal in this January, this month. A highly durable RNAi therapeutic inhibitor of PCSK9. Well, you look at that title and you think, I don't want anything to do with this. But it turns out that PCSK9 is a protein that has to do with low density lipoprotein, and you can't really get your LDL to work very well if you don't have this. So if you get this inclisiran, which is the special iRNA that you can infuse, or that you can infuse, you give it as an injection, it will lower your low density lipoprotein by 50% or more, and one injection lasts 6 months. That's pretty cool. The CRISPR, which is even more exciting, is very similar in that you're using it stands for clustered regularly interspaced short palindromic repeats. You can just kind of toss that off. And CRISPR is easier. But it's a ribonuclear protein. It's been used extensively in mice. You design the thing to latch onto a certain part of the DNA you want to clip out, and then you can clip out that part of the DNA, and you can put, and you actually can put in segments that are, instead of being defective, are corrected, so that you're correcting a gene defect. If you did this very early on in development, you could actually have a child that was quite normal and would pass on that normal DNA. But that's one problem with it, is to get there early enough. But right now, just using it in adults, there's a clinical trial in humans that's been started in China, so that's underway. And right now, there's work underway at Stanford and Berkeley to excise the mutation in sickle cell disease and put in a correct DNA for the beta globulin that is a problem in sickle cell. And the NIH has already approved a clinical trial in the U.S. to edit T cells for cancer therapy this year. So this you can find if you look at the very recent Science magazine from this month. There's a section about products in the back, 207 and 209, I think this is. And it just talks a little bit about this and what actually happens. But there was a recent article in the Wall Street Journal about CRISPR, and they said that any idiot can do it. And they had the author decided that he could be the idiot, so he went to a lab and found it was a little harder than that, but not much. So it's going to be a therapy that we're going to be using one way or another fairly soon. So that's what I had to say. I'm happy to, I'm 2 minutes over, but I'm happy to answer questions if I can. Yes.
Steve, if we have FSH patients